from the clinical data also today of patients with an islet transplant obtained the data via the International Transplant Registry on the one hand and from single cases on the other hand. But whether already the time has come to draw general definite conclusions for the further use of islet transplantation, this is a question which has been asked by many colleagues. So the program committee has planned to make the attempt of such an open seminar chaired by Dr. Potzer and myself with those colleagues working actively with islet transplantation who are asked to express their opinion to a couple of questions, if possible, and to give perhaps emphasis to one or the other topic. This within 10 minutes, if possible, for each colleague. Later on, then, should be time for questions from the floor. The colleagues are Dr. Scharp from St. Louis, Dr. London from Leicester, Dr. Zocchi from Milan, and Dr. Hering from Giessen. Now, the topics we have put together were present indications for islet transplantation and perhaps future, the choice of metabolic parameters prior to and after islet transplantation, the conditions for donor and donor glands which should be fulfilled, and finally the choice of immunosuppressive treatment prior to transplantation and afterwards. Now, all colleagues will give their statements or their opinions in the time, perhaps to all of the four questions, but perhaps also with emphasis to one or the other. Maybe start with Dr. Scharp from St. Louis, who is a pioneer in islet transplantation in humans, and he will try to outline his personal opinion.
Thank you, Professor Federlin. If I can have the first slides, please. Being the opportunity of the first speaker for the tasks outline, I thought I would just introduce a few items that I think are critical to those of us looking at clinical islet transplantation. And looking at the procedures involved in this step, or this effort, they really fall in these four areas of pancreas procurement, of course, islet isolation and purification, islet preservation, and transplant, which is the simplest, especially in the portal injection. Next slide. The procedures, we spent a lot of time talking about collagenase digestion of islet isolation, purification, and I think this workshop, again, has benefited us in terms of being able to discuss in this kind of atmosphere. Especially, I think the critical problem that relates to standardized collagenase, looking at what are its critical components, how do we control the process to optimize the digestion and freedom of islets, what may be the role of inhibitors in this process, and then what's the role of the other problem, which is the pancreas itself and its own endogenous enzymes. And I think that we spent a lot of time in our workshop last night on collagenase, that these are critical problems if we expect to have large-scale clinical application. We have to solve these problems before we can go much further. Next slide. The other process towards large-scale clinical devices, or large-scale clinical processing of islets, is the dedicated processing facility. And I wanted to share with you that in St. Louis, we set this up in May at the funding of our hospital. This is one of 5 modules which are all linked together for the processing. The pancreas is presented in here. In other words, we've escaped from the laminar flow room where we've been trapped for the last several years. And this situation now is providing excellent sterility and ease of processing in terms of setting up a controlled environment that will lead us down the road to what will happen in the States in the near future, which is FDA control of this process. Next slide. Certain quality controls, and we've talked about these in other groups, are critical as we think about moving in the clinical and working islet viability, we've just heard papers relating to these. Next slide. Islet sterility, we've talked about that, processing laboratory areas. But if we get now to the questions that we're addressing, what really are our specific aims in terms of clinical islet transplantation? And I suggest that if we divide our aims into immunosuppressed patients, those studies we were looking at immunoisolation, and those studies looking at immunoalteration, we're really at the state where we're looking at islet dosage preservation, effective immunosuppression, and islet function. And I think the four topics selected for our discussion address these areas very well. And this will be the mainstay of numbers of patients in this area. But there are preliminary studies developing in these two areas, which are basically aiming toward the development of clinically relevant devices and the feasibility of clinical application of what's worked in rodents. Next slide. And I wanted to share with you just the latest patients because of improving in the processing, the new facility in St. Louis and work in the collagenation. We've transplanted 4 patients, and this patient has now been insulin independent for 7 weeks. And I just wanted to briefly go over his data. This patient in the blue line, sugars here, C-peptides here, was prior to any transplantation in 1989. He had a subtherapeutic islet transplant in 1989, which was followed and essentially failed. And this is his glucose sustacal before, and C-peptide. He had a little bit of marginal C-peptide before the latest transplant. At 1, 2, and 3 months, his glucose response to sustacal challenge while on intensive insulin therapy showed 1 month, 2 months, and 3 months. And I note that these are picomoles per mil, not nanogram levels, showing very good responsiveness. And at 3 months, when his prednisone came down to 0, we took him off insulin. And here's his glucose response a month later. He's now 7 weeks out. And C-peptide levels in this range. This patient is the first of 2 patients that are the beginning of our islet dosage study. And this patient received 17,000 islets per kilogram body weight in what is with multiple donors, 4 donors. And is, I think, partly responsible for achieving these kind of levels. Next slide. The use of the sustacal stimulation index shows all 4 patients. This patient in blue received only 8,000 islets per kilogram body weight, was not able to achieve insulin independence. This is the patient who is insulin independent at the time of insulin independence here, and then 4 months later is being followed. This patient had islets in similar ranges here, but underwent rejection episodes of his kidney at 2 weeks, 3 weeks, and 4 weeks, and has since lost function. This patient was just transplanted 3 weeks ago. So these kinds of data, I think, suggest that this is just an example. If I have lights, please, I'll just leave the lights off. I'll move over there. These kinds of data then suggest that indeed we can develop indications for clinical trials. But before looking at what the indications, I think some comments about what our objectives of clinical trials should be. And I would suggest for our discussion that in the immunosuppressed patients, what is the optimal islet dosage? What is the optimal effect of immunosuppression? What problems do we have in preservation? Because we know that preserving islets in culture for up to 7 days clearly results in problems with loss of islet mass in terms of eventual transplantation. But we think, based on rodent studies, that especially room temperature culture combined with induction therapy may reduce the risk of rejection episode. So what are the real clinical facts and data based in patients that say it's worth culturing versus reduction of rejection episodes? We've heard at this meeting the potential importance of growth factors. For example, nicotinamide given to patients for a year. Does that explain explain the major difference in our center where we have patients with almost enough to become insulin independent early but not enough, failing over time, versus the difference in the Giessen group where now you have patients who have almost had a marginal amount who now have gotten better. I think it's a very exciting potential. So to me, these are the objectives of immunosuppressed clinical trials, looking at outcomes of well-documented insulin independence, rejection episodes, and duration of function. And the bottom line really is, can we establish this as a clinical utility, especially with the knowledge of the DCCT trials. The other set of patients that are undergoing clinical trials fall in two other groups, those with immunoisolation and those with immunoalteration. These are very more futuristic kinds of activities and only taking place a few places. But I would suggest that the objectives in immunoisolation, first of all, a proof of principle kind of trials, and I'll be presenting data from our center later at the next session this afternoon of such a trial. But then probably a series of device development trials looking at device design and inner function of subtherapeutic amounts of islet tissue. What's the effect of xenografts in clinical trials? To finally develop a clinically relevant device that has therapeutic efficacy and safety. Then finally, immunoalteration studies looking more futuristic because until this meeting I've not seen any data in anything but rodents, but we had one presentation from the Cambridge group in dogs at this meeting of the immunoalteration regimen. And I suggest to you that a model that we're working, just starting now, is, and this paper is the humanized skid mouse model. That's not a new idea, but what has happened is through collaborators at our center is about an 80% success rate of developing humanized skid mouse with disseminated human immune cell function. And this paper has just been accepted for transplantation, and I'll be happy to send people a preliminary report of those if you're interested. But the reason we developed this model, or tried to develop this model, is because how do you decide what 30 or so different ways of immunoalterations that may work in rodents do you find any kind of clinical application? So what we're going to be doing is trying to develop these in a systematic way to develop ones that may have clinical utility. So in closing then, I would suggest the following indications for clinical islet transplantation. Those in the simultaneous kidney islet transplant, again based on our clinical objectives and outcome measurements, would be in two groups, those getting kidney islets under standard induction immunosuppression, somewhere between 0 to 7 days of the kidney transplant, while that induction therapy is still important in terms of allowing graft acceptance. In the established kidney islet, or the islet after kidney, I have to get used to changing the term, islet after kidney term, is probably the more effective term. What's the role of maintenance immunosuppression, new induction immunosuppression? And we've been using at least 6 months of stable kidney function before approaching these patients. But that's a suggested starting point. And again, there'll be very special, focused, limited trials that we've discussed for immunoisolation, device development, and immunoalteration.